attack arrived without a sound. One moment the platform was ordinary, commuters checking phones, trains sliding into the station, and the next, the air carried something that the body could not expel. In emergency rooms across Tokyo, doctors watched men and women arrive conscious but unable to control their own muscles, pupils pinned to specks, lungs filling with fluid, the body's signals telling it to contract and never release. The weapon was invisible, nearly odorless, and its makers were not a foreign army but a religious cult with a laboratory in a concrete building north of the city. To trace how a molecule born in a German laboratory in 1938 ended up in the hands of an apocalyptic sect, we leave the bright surface of that morning and descend into the sealed archive, where the hatches close, the pressure builds, and the files wait in the dark. This is the Deep Archive. Inside, you will find a glass vial from a Nazi inventor's bench, a shipment of dismantled machinery that crossed the Atlantic to be reassembled in the Colorado desert, a Utah valley where thousands of sheep fell dead from a drifting chemical cloud, a human test subject inside a Porton-down gas chamber, the Kurdish town of Halabja, where Sarin poisoned the genetic line of families yet unborn, and a forensic chromatograph in Tokyo that read the impurity in a single drop of liquid like a written confession. The archive opens with the doctor who saw the invisible, a man who had trained for a war that never happened, and then recognized a weapon that no one had declared. This is Sarin, the cult that brought nerve gas underground. The fluorescent lights in the emergency corridor of St. Luke's International Hospital did not flicker that morning. They held steady and cold, washing the polished linoleum in a flat institutional white that made every shadow disappear. No one had planned for shadows on a Monday in March, when the morning was clear and the air outside held the first quiet hints of spring. The automatic doors opened at 8:22 a.m. with their usual soft sigh, and Dr. Nobuo Yanagisawa stepped through them into a day that had not yet revealed itself. The night shift had been quiet. The charge nurse reported 3 admissions after 2 a.m., all stable. The cardiac unit was expecting a transfer at 9. The neurology department had 1 patient scheduled for discharge, an elderly man whose left hand had regained enough strength to grip a spoon after 6 months of therapy. It was the kind of morning that allowed a doctor to think about lecture notes and the small corrections he wanted to make to a paper on proximal myopathy. He turned the corner toward the central desk, and the smell he caught was not the cleaner solvent. It was a thinner, sharper note, a chemical whisper that tightened the back of his throat in a way he could not immediately explain. He saw the charge nurse before she saw him. Her face was not the face of someone managing a routine Monday. It was tight and pale, the face of a person who had been holding her breath without knowing it. Before he could speak, a man in a gray business suit pushed through the entrance, bent double. His legs folded without warning, as though the bones had turned to hot wax, and he collapsed onto his knees on the polished floor. He did not cry out. He simply fell forward, his chest heaving, his face a mask of total inward focus. Yanagisawa dropped his briefcase and moved toward him. The man's breathing was a wet, strangled sound, the sound of lungs laboring against a tide they could no longer clear. When the doctor looked into his eyes, the pupils were no larger than a pencil point, dark specks drowning in the brilliant corridor light. The skin around them was dry, but a thin, stringy line of saliva threaded from the corner of his mouth to his collar. The man's briefcase was still clutched in his right hand, and Yanagisawa had to pry the fingers open. The handle was slick with a faint, strange residue that left no visible mark. Yanagisawa had seen pupils like that in only one place before: the chemical warfare manuals he had studied years earlier, when the medical curriculum still included a chapter on nerve agents born from the Cold War's secret arsenals. The manuals described a substance that could reduce a man's pupils to pinpoints in seconds, a substance that had never been used in war on Japanese soil, and certainly never during a morning commute in Tokyo. His first conscious thought was not a diagnosis. It was the quiet, professional refusal to believe what his training was telling him. Organophosphate poisoning did not happen here. Within the next 30 minutes, the emergency room filled with other men and women wearing the same gray suits and the same impossible symptoms. They arrived in waves, carried by colleagues or pulled from the back seats of cars that had halted mid-journey in the streets outside. Some walked in on their own legs, only to crumple against the admitting desk as soon as they stopped moving. Others were brought on improvised stretchers of ripped fabric, their bodies limp except for the occasional violent muscle spasm that writhed through their limbs. None of them had a wound. No one had blood on their clothes. The injury was entirely inside them. For the people on the trains, the first warning had often been blurred vision. The world went dark at the edges, then narrowed to a tunnel, then a pinhole. A woman would look at her own hand and see it as through a straw. Then the difficulty breathing began, not a constriction of the chest, but a flooding from within, as though the lungs were filling with a substance the body was producing too fast to clear. By the time they understood something was wrong, their legs would no longer hold them, and they would slide to the floor of the carriage amidst the briefcases and newspapers of a working Monday morning. The symptoms were those of a nerve agent, a biological override that Yanagisawa knew only from Cold War manuals. It was a chemical hijacking that occurred in the space of a single breath. It was not a disease that evolved over hours. It was a chemical hijacking that occurred in the space of a single breath. The antidote, when it existed, was a pair of drugs: atropine, an ancient extract from the belladonna plant that could dry secretions and force the pupils to widen, and pralidoxime, a synthetic molecule engineered during the Cold War to chemically pry the poison from the nerve endings. Given early and in volume, they could reverse the process, slowly, partially, with patients often requiring repeated injections for hours. The medical manuals had recommended a dose of atropine 20 times the normal therapeutic limit, a quantity that would have been fatal under other circumstances, but was the only barrier between the patient and respiratory failure. The pharmacy at St. Luke's held a standard hospital store of atropine, enough for a year of surgical emergencies. 40 minutes into the crisis, the last ampule was being drawn into a syringe. Additional vials of PAM were rushed from a nearby branch hospital, but the stream of patients outpaced every delivery. The vials emptied faster than runners could carry them. The syringes were reused under pressure. Every protocol said they should not be. The hospital's neonatology ward sent its only mobile ventilator down to the emergency floor. The surgical team, in between their scheduled procedures, began managing airways on gurneys in the hall. The cafeteria stopped serving food and became a triage area for patients who were still breathing on their own. The vending machines sold out of bottled water as the staff sweated through the morning, their own throats raw from the faint chemical residue that clung to the patient's clothes. The military directed additional PM to the hospital from its emergency stores. It arrived by helicopter at mid-morning, and the vials were run down 6 flights of stairs because the elevators were too slow. By noon, the number of those affected had passed 5,000. The official count would later settle at 13 dead and over 5,800 injured. But numbers on a page could not capture the manner of the dying. The people who lost control of their breathing in the tunnel, on the platform, in the back of a taxi that would never reach the ward. Yanagisawa moved through the rooms as the morning became afternoon, working muscle memory he had never expected to use. He had trained for this in a world where such attacks were imaginable only as military exercises, scenarios drawn from a 1960s fear of a Soviet incineration that never came. The textbooks had names for the agents: Tabun, Sarin, Soman. They were listed with the chemical formulas he had memorized and then set aside, because no Japanese civilian would ever need to recognize them. But the scripts of war have a way of arriving in peacetime, and they demand that the doctor becomes something between a physician and a field medic in a conflict without borders. The telephone lines to the hospital jammed by mid-morning, and the city's emergency services switchboard collapsed under the weight of calls. For a time, the entire medical network operated on rumor and radio. The dispatchers calling out station names like a list of battlefields: Kasumigaseki, Kodamacho, Kamiacho, Naka, Meguro. The spring morning outside the windows remained bright and indifferent, the sky a clean blue that no smoke had marked. When the last of the day's patients had been stabilized or moved to the quiet room where the dead awaited identification, the hospital corridors fell into a strange, suspended silence. The fluorescent lights still burned, relentless and unchanging. The air smelled of disinfectant and a faint, sweet chemical residue that no amount of cleaning could erase. A staff member walking along one of the affected platforms at Kasumigaseki Station noticed a sodden plastic lunchbox lying against the rail. Its lid was cracked, and the contents, a neat arrangement of rice and pickled plum, had spilled across the concrete, trodden into a gray paste by the boots of rescuers. The lunchbox itself was wet, not with water, but with a clear, faintly oily liquid that had pooled beneath it. The liquid had no smell. It left no stain that the eye could detect. It was just a sheen on the plastic, a dampness that did not evaporate. Decades earlier, a chemist in a German laboratory had recorded the formula for a compound that he initially called Substance 146. He was not thinking about Tokyo or subways or lunchboxes. He was thinking about pests and crops and the hopeful, brutal logic of agriculture. But chemistry is indifferent to intention. The molecule he made was a key that fit the lock of the human synapse, and once turned, it could not be withdrawn alone. The liquid on the lunchbox carried the signature of that original synthesis, a molecular lineage that had traveled from a quiet European laboratory into the arsenals of empires, and from those arsenals into the hidden workshops of a cult that believed the end of the world was imminent. But in the emergency corridor of St. Luke's on that March evening, none of that was yet known. Dr. Yanagisawa stood under the steady lights, his coat stained with the day's fluids, and understood only that he had witnessed a weapon that no one had declared, from a source no one could name, rising out of the most ordinary moment of a city's life. The molecule had a history, and that history was still locked behind doors that the doctor could not yet open. The liquid on the lunchbox was the latest entry in a chronicle that had begun far away, and the next entry would be written by those who traced it back to its source. In 1993, 5 years after the planes passed over Halabja, a different kind of laboratory had begun its work on the far side of the continent. In the cedar forest of Kamikuishiki, where the shadow of Mount Fuji fell across the hills, a cluster of huge white silos rose from the undergrowth like a misplaced industrial abscess. The hum of circulation pumps carried through the trees, blending with the low, rhythmic chant of acolytes in a nearby meditation hall. The air smelled of pine and, faintly, of something sharper, a solvent note that did not belong to the forest. This was the compound of Aum Shinrikyo, the supreme truth, a religious movement that had grown from a small yoga school in the Tokyo suburbs into an empire of apocalyptic belief and enormous wealth. And at its scientific heart was a man named Saiichi Endo. Endo was not a chemist by training, but a physicist, a graduate of the University of Tokyo who had studied fluid dynamics before drifting into a succession of odd jobs and spiritual searching. In his late 20s, he encountered the teachings of Shoko Asahara, the nearly blind, charismatic founder of Aum, who preached that the world was careening toward a final war, a catastrophe that would kill everyone except the enlightened few. Endo found in Asahara's prophecies a structure that his physics textbooks could not offer, a purpose. He was named the cult's Minister of Science, a title that sounded like bureaucratic satire but carried absolute authority over a growing technical staff. By 1993, Endo supervised a project that no non-state group had ever attempted, the independent manufacture of the nerve agent Sarin. The origin of that project lay in a logic as old as the concept of holy war. Asahara, convinced that the Japanese government and the United States would soon attack his movement, ordered a crash program to develop weapons of mass destruction. The cult had already tried to buy assault rifles from the Russian mafia and sought nuclear warheads from the collapsing Soviet Union. But what came to hand most easily was chemistry. Endo's team began by studying publicly available textbooks, military manuals, and patent filings from the 1930s, the same roadmap that the German Reich had used 50 years earlier. They understood early on that the key was not theoretical knowledge, which was already in the open literature, but the engineering of a safe production line. The trick, as one intelligence analyst later put it, was not learning to make sarin, it was learning not to die while making it. The laboratory they built sat inside a large warehouse-like building hidden from the road. Inside, stainless steel reactor vessels rose 2 stories high, connected by a lattice of pipes, glass condensers, and vacuum lines. Large double-pane windows separated the control room from the production floor, and closed-circuit cameras monitored the critical stages. The staff wore full-face respirators and chemical suits imported from Russia, another acquisition that was entirely legal at the time. When the first batches were synthesized, the liquid came out pale yellow, carrying an odor that one survivor described as a cross between turpentine and burnt plastic. It was sarin, but crude, marred by byproducts that the Reich's industrial chemists would have considered a failure of quality control. Yet it was potent. A single droplet the size of a match head could, in theory, arrest the breathing of an adult man within minutes. The work was dangerous, and the compound kept its own cost. Several members of Endo's team were accidentally exposed to vapor leaks. They collapsed in classic organophosphate distress. The cult's medical unit recognized the pattern immediately from military manuals, even if the Tokyo hospitals would not have placed it. The cult's medical unit, which ran a hospital in Tokyo, treated them in secrecy, administering atropine and prolidoxime, the same antidotes used at Porton Down and Edgewood. None of them went to a public emergency room. The injuries were logged as private religious retreats. The strange part was not that the accidents happened, but that they were treated with such clinical precision by a group that believed in reincarnation and levitation. In Kamikui Shiki, the spiritual and the technical had merged into a single terrible competence. The theology that anointed this work was a distortion of Tibetan Buddhism mixed with science fiction and conspiracy theory. Asahara taught that the coming Armageddon was prophesied by Nostradamus and could be accelerated by the righteous. The killing of enemies was not murder but an act of poa, a tantric ritual that liberated the soul of the victim from bad karma. Endo himself wrote internal documents that framed chemical weapons as a form of mercy, a swift transition to a higher rebirth. Under this logic, the Sarin factory was not a death plant, but a sacred machine, its pumps a kind of prayer. The low chanting of the acolytes that mixed with the industrial hum was not incidental, it was part of the production cycle. By the spring of 1994, Endo's team had produced enough liquid sarin to fill a small fuel truck. The cult now needed a real-world test. The target chosen was Matsumoto, a city in the Japanese Alps, where a panel of judges was presiding over a land dispute that threatened Aum's real estate holdings. On the night of June 27, a specially outfitted refrigeration truck drove into a quiet residential neighborhood near the judges' homes. The vehicle carried a steel tank, a heater, and a powerful fan. At around 11 o'clock, the operators opened the valve and began to vaporize the sarin into the summer air. The gas spread in an invisible plume, pushed by the night breeze into an apartment complex and along a narrow street. Within minutes, the first residents were collapsing in ways the local emergency rooms could not explain. An 81-year-old man collapsed on his doorstep, unable to breathe. By the time the hospitals began to suspect a chemical agent in their midst, 7 people were dead. Over 600 sought treatment. The staff worked through the night, uncertain of what they were facing. The police investigated but found no industrial accident, no factory leak, no transport spill. The case went cold. Aum Shinrikyo had detonated the most guarded chemical secret of the 20th century in a Japanese suburb, and nobody recognized it for what it was. Endo and his colleagues studied the Matsumoto data like any research group analyzing an experiment. The sarin had worked, but the dispersal had been uneven. The wind had spared some buildings entirely while saturating others. The cult concluded that the next delivery system should be an enclosed space, a tunnel, a train, where the gas would linger and the atmospheric variables could be controlled. They began refining the binary mechanism, keeping the chemical precursors separate until deployment, a technique they had read about in declassified American patents. The Kamikuishiki compound hummed with renewed intensity. The silence that followed Matsumoto was not just a failure of detection, it was an unwitting permission. Every month that passed without an arrest confirmed Asahara's conviction that the state was blind and that the prophecy could be fulfilled. By the end of the year, Endo's laboratory had produced a more concentrated, more lethal grade of sarin. The storage tanks were full. The delivery teams were trained. The weapon of the Cold War superpowers was now an article of faith for an apocalyptic cult. Back in the cedar forest, the white silos caught the morning light. The pumps continued their steady, indifferent pulse. The meditation halls filled with the sound of sutras. The strange marriage of incense and solvent hung in the air. And inside the warehouse, a physicist who had once studied fluid dynamics at the finest university in Japan was writing the final specifications for an attack that he believed would hasten the salvation of the world. The molecule had no doctrine, but it had found a congregation. The train line waited exactly where it had always been. The umbrellas were the first thing the evidence technicians noticed. Not because they were rare, Tokyo in early spring was a city of umbrellas, but because the tips had been ground to a hard point, then wrapped in cloth and sealed inside clear plastic evidence bags. The plastic was fogged with the last moisture of the subway air, the same air that had carried something that morning that no one could see. In a laboratory on the other side of the city, a forensic chemist named Masayuki Nagao prepared to receive a different order of evidence. He was expecting a set of glass vials, each containing a few milliliters of liquid recovered from the floor of a train car, soaked into newspapers, and transferred by investigators in full protective suits. It was 4 days after the attack that had turned Tokyo's underground into a silent emergency room, and the city was still holding its breath. The facts of what happened were already becoming clear. That morning, 5 men had boarded 5 separate subway lines during the rush hour. Each carried a plastic bag filled with a clear liquid that looked like water. At a prearranged signal, they dropped the bags, punctured them with the sharpened umbrella tips, and stepped off the trains. The liquid spread into the air and onto the car floors. Within minutes, the stations became spaces of collapsed bodies and stifled breathing. But the police, who had already been investigating a religious cult for a previous chemical incident, had a suspect. What they needed was proof, a molecular chain that could not be argued away before a judge. Nagao was a meticulous man, trained in analytical chemistry, the craft of teasing out a sample's hidden composition. He began by feeding a few microliters of the liquid into a gas chromatograph. The chromatograph produced a jagged line on a screen, a series of peaks, each representing a different compound. The largest peak was unmistakable: isopropyl methylphosphanofluoridate, the chemical name for sarin. But Nagao's eye went to the smaller peaks that surrounded it. They were not supposed to be there. In a military-grade synthesis, sarin emerges pure, its chemical surround silent. The reaction sequence is controlled so tightly that the final product contains only the intended molecule. But the sarin that Nagao was looking at was crowded with chemical neighbors: alkyl phosphonic acids, isopropyl esters, and traces of unreacted precursors. These were byproducts of a hasty, imperfect reaction, the molecular equivalent of a rough draft. The presence of these compounds told a story that a pure sample could never tell. It was the story of a laboratory that had learned the chemistry, but not the craft. The cult's production facility, which investigators had raided just 2 days after the attack, had been an industrial parody of a chemical plant. Inside, they found glass reaction vessels, heating mantles, and rows of plastic containers labeled with the names of common industrial chemicals, not the tightly controlled precursors a military program would use, but substances available through ordinary commercial channels. The cult's chemists had followed a synthesis route published in open scientific literature, a route that a determined student could replicate with a few $1,000s and a well-vented workspace. But their equipment was basic, their temperature controls imprecise, and their stirring inconsistent. The result was a sarin that was about 60% pure, the rest a soup of side products. It was a weapon, but it was also a messy artifact. Each byproduct told a specific story. Water crept in at the wrong moment and split unfinished molecules. A sulfur residue betrayed the solvent the cult had used. Heat damaged intermediates revealed a rushed, uncontrolled cascade. Together, these impurities formed a unique profile, a chemical fingerprint that no other batch in the world could duplicate. Nagao realized that this fingerprint could be compared to samples taken from the cult's hidden laboratory. When he ran the laboratory residues through the same chromatographic analysis, The pattern of peaks was a near-perfect match. The same unexpected byproducts appeared in the same ratios. It was as if the sarin in the train cars and the sarin in the Colt's vats shared a genetic code. The match was so precise that it eliminated any chance that the weapon had come from another source. No other known production run in the world had generated that exact suite of impurities, and the odds of a random coincidence were beyond calculation. For Nagao, the moment of confirmation came not as a flash of revelation, but as a slow settling of certainty. He had spent days staring at the peaks, comparing their retention times, running control samples from unrelated chemical reactions to exclude contamination. The pattern was not only consistent, it was explanatory. It told him that the attack was not a random act of terror, but the culmination of a deliberate, if flawed, industrial effort. The molecule was, in effect, a confession. But the story went deeper still. As Nagao and his team traced the byproducts back through the synthesis pathway, they found themselves looking at the ghost of an older chemistry. The route the Colt had used was a modified version of the original process developed in the 1930s by the German researchers who first stumbled onto nerve agents. Those German chemists, working with limited equipment and impure starting materials, had recorded the same side reactions in their laboratory notebooks. The Colt had not just recreated the weapon, they had recreated the historical accidents of its birth. The molecule was a palimpsest, and under the surface of the Colt's sloppy batch, the handwriting of the original inventors was still legible. The trial of the Colt's leadership began in April 1996. The courtroom was a space of fluorescent light and wooden benches, packed with survivors and journalists. The prosecution presented Nagao's findings as the scientific spine of its case. Large charts showing the chromatograms were displayed on easels, the peaks marked with arrows. The defense argued that the evidence was circumstantial, that the sarin might have been planted, that the matching impurities could have occurred by chance. But Nagao's testimony was unshakable. He demonstrated that the probability of an identical byproduct profile arising from an unrelated synthesis was astronomically small, smaller than the chance of a fingerprint being shared by two strangers. The impurity was the signature, and it signed the Colt's name. One by one, the Colt's inner circle was convicted. The guru who had prophesied the apocalypse and ordered the attack, the engineer who had designed the production line, the chemist who had pored over textbooks in a concrete bunker, his hands trembling from the tension of the final synthesis step. The sentences were death or life in prison. The verdicts relied on many pieces of evidence, financial records, witness testimonies, the physical layout of the laboratory, but the chemical evidence was the one that could not be cross-examined. Molecules do not lie, and they do not forget. Nagao rarely discussed the case in public after the trial ended. He returned to his laboratory, where he continued to develop forensic methods for detecting chemical agents at ever lower concentrations. The techniques he pioneered became standard in the field, used later by United Nations investigators in Syria and by response teams after assassinations. He became, in a sense, the archivist of the invisible, the man who taught the world how to read the molecular traces of atrocity. The Chemical Weapons Convention, signed in 1993, entered into legal effect on April 29, 1997, after the Tokyo trial pushed a wave of nations to ratify. Sarin was listed as a Schedule I substance, a chemical with no legitimate peaceful application. but the convention's verification machinery was designed for declared state stockpiles and scheduled destruction. A religious cult with a concealed laboratory and a well-funded library card fit none of its categories. But the impurity that convicted them was more than a signature. It was a map. The byproducts Nagao identified pointed backward to a specific lineage of organophosphate synthesis that had its origin not in a cult's concrete laboratory north of Tokyo, but in a German greenhouse in the spring of 1938. That is where the molecule began its journey. In the hands of a chemist who believed he was looking for a better pesticide. The laboratory at Leverkusen was a large, high-ceilinged room on the second floor of Building 15, its frosted windows throwing a cold, even light over the long benches. It smelled of bitter almonds that morning, a faint, sweet backnote beneath the sharper reek of ether and the mineral silence of cleaned glass. Gerhard Schrader leaned into the brass microscope lamp, adjusting the beam until it fell exactly on the row of 5 ground glass vials he had prepared the night before. The fourth vial held a colorless, nearly odorless liquid that he had designated only by its code, GB. It was the spring of 1938, and Schrader, a methodical 40-year-old employee of IG Farben, believed he was still searching for a better way to kill aphids and mites. He had been searching for that for nearly a decade. Born in 1903, Schrader had studied chemistry at Braunschweig and joined IG Farben's pesticide research division in the early 1930s, when the world's food supply depended on heavy metal poisons. Arsenic, copper, mercury spread thick over the fields. The promise of synthetic organic chemistry was that it could replace those crude, soil-poisoning compounds with precise molecular tools, molecules that would enter the body of a specific pest, disrupt a specific reaction, and leave the warm-blooded farmer unharmed. This was the logic of organophosphates, a family of phosphorus-based esters that Schrader began to explore with the quiet obsession of a man who loved the clean lines of a structural formula. So the first serious warning came not as a failure, but as an over-success. 2 years earlier, in 1936, Schrader had synthesized a compound called tabun. In the first insect screening, it killed everything in the test cage within minutes. But when a tiny droplet accidentally touched the skin of a laboratory assistant, the effects were immediate and terrifying. The light in his assistant's eyes narrowed to dark, drowning specks. He began sweating profusely, breath irregular. The emergency team had no diagnosis that fit. He survived only because the dose had been minuscule, and because Schrader by then understood that atropine, a drug that blocked the overstimulated receptors, could act as a temporary wedge against the poison. The military was informed. IG Farben's directorship classified tabun as a war gas, and Schrader was ordered to refine the molecule further to see how deep the lethality went. So by the spring of 1938, when the light of that brass lamp fell on the fourth vial, the objective had already shifted. Schrader was no longer looking for a selective insecticide. He was mapping the boundaries of a new kind of weapon. The liquid in the vial was a second-generation organophosphate, chemically similar to tabun but far more stable, and when he tested it on laboratory animals, far more potent. The numbers that came back from the biological assays were difficult to believe. A concentration of 1 part per million in air could kill a monkey in less than a minute. A droplet too small to see could fell an adult human. The new compound was approximately 500 times more toxic than tabun. Schrader named it, eventually, after the first letters of the 4 men most directly involved in its evaluation: Schrader himself, Otto Ambros, Hans Rudiger, and a chemist named van der Linde. The combination formed the word sarin. Schrader understood the mechanism because he had mapped it. The body's every motion, breath, heartbeat, the curl of a finger, depends on a molecule called acetylcholine, which carries the signal from nerve to muscle and is then instantly shredded by an enzyme, acetylcholinesterase, to stop the signal. Schrader's compound slipped into that enzyme's active crevasse and formed a bond that could not be broken. The enzyme was not simply blocked, it was destroyed. One molecule of sarin could silence one enzyme molecule forever, and each enzyme could have processed thousands of signals per second. The cleanup crew never arrived. Acetylcholine flooded the synapse without pause, and the mechanical sequence of collapse began. A standard assay from Schrader's own laboratory notes recorded prostration, convulsions, respiratory arrest at 45 seconds. The paradox was not merely physiological, it was historical. A man who had set out to protect the garden had invented a substance so lethally optimized that it had no conceivable agricultural application. Unlike Tabun, which still had some structural cousins that could be diluted into insecticides, sarin was pure weapon. Its volatility, its stability in storage, its ability to kill through both inhalation and skin contact, its utter lack of warning properties, odorless, tasteless, invisible, made it an ideal chemical for war. The Nazi regime, through IG Farben, immediately classified it as a state secret, sequestered the research behind armed guards, and began to plan for industrial production at a dedicated site hidden in the forests of Silesia. But even before the first factory was built, the molecule itself was already a fait accompli. It existed. It could not be uninvented. Schrader himself remained an elusive figure in this history. His post-war testimony, given to Allied interrogators, was characterized by a technician's precision and a noticeable emotional distance. He spoke of yields and syntheses, not of consequences. One interviewer noted that Schrader seemed to regard the molecule as a problem solved, a chemical achievement that stood on its own, independent of its use. That detachment, whether honest or protective, would become its own kind of signature. The inventor who, having delivered the horror, retreated into the safety of the laboratory notebook, the neat columns of data, the clean light on the bench. But the liquid in the fourth vial did not care about its inventor's intentions. It had no politics, no memory, no remorse. It was simply a configuration of carbon, hydrogen, oxygen, fluorine, and phosphorus. A small, stable arrangement of atoms that happened to fit perfectly into a critical biological lock. The vial itself was small enough to disappear into a coat pocket. The amount needed to kill a roomful of people could be measured in grams. In the coming decades, that portability would rewrite the rules of deterrence, of terrorism, of public trust in the air one breathes on a morning commute. But on that spring day in 1938, all of that lay in the future, sealed in a laboratory safe under the hum of a ventilation fan, waiting. The light on the bench threw the shadow of the vials onto the dark wood. The almond scent had long since dissipated, replaced by the neutral, almost cold emptiness of a room where a new thing had come into the world. Gerhard Schrader washed his hands at the stone sink, dried them on a bleached towel, and wrote the next entry in his laboratory journal. The entry recorded the reaction conditions, the yield, the biological assay results. It did not record the question that would define the rest of the century. Once a molecule like this exists, who decides when and where it is used? And what happens when that decision falls into hands that do not share the rules of the laboratory? The first Soviet soldier to push open the steel doors at the Dehernfurth plant felt the cold before he registered the smell. The factory had been deserted for less than 24 hours, but the boiler still breathed a low residual heat into the galleries. Overhead, the pipework stretched like a glass and iron arterial system, sheathed in hoarfrost where the steam had condensed and frozen. He walked past steel vats the height of a grain silo, past control rooms where the brass dials still trembled at the bottom of their scales, measuring pressures that no longer existed. A side corridor led to a row of doors sealed with fresh paint, each bearing a single yellow stenciled skull. The silence was not the emptiness of abandonment. It was the peculiar silence of a machine paused mid-cycle. All the pistons at rest, all the valves shut, all the chemistry suspended in expectation. What he did not know, what no one in his regiment could know, was that he was standing in a plant that had produced several thousand tons of a liquid weapon unlike any in recorded history. A substance that could kill with a single droplet on exposed skin, that traveled through the air in an invisible, almost odorless vapor, that turned the body's own chemical signals into an unending, fatal paralysis. The Soviets sealed the site and sent its surviving documents east by train to a Moscow institute where a separate team of chemists would spend years reconstructing the formula. But the plant itself, the furnaces, the glass-lined autoclaves, the fractionation towers, would soon become the prize in a hunt that spanned a ruined continent. Within weeks of the German surrender, a quiet operation began under the innocuous name of the Combined Intelligence Objectives Subcommittee. British and American chemical warfare officers, many of them young men with freshly minted PhDs, fanned out through the occupation zones clutching lists of industrial installations. They were chasing not just the machines, but the men who had run them. Interrogations of captured engineers revealed the true scale of the nerve agent program: 2 major production sites, thousands of tons of precursor chemicals, and the pilot-scale output of a third variant even more persistent than the first. The material they sought was concentrated at Dehernfurth and a sister facility, but the key personnel had already slipped westward ahead of the Soviet advance. They were located in rented rooms, in farmhouses, in the makeshift holding pens of prisoner-of-war camps. A few presented themselves at checkpoints, offering expertise in exchange for a clean slate. The arrangement acquired a flat bureaucratic title: Operation Paperclip. Its moral arithmetic was brutally simple. The Soviet Union now held the physical factory. They had the blueprints. They possessed residual stocks of the intermediate chemicals. What they did not yet have, and what the West would deny them for as long as possible, was the unwritten knowledge, the temperature at which the fluorination reaction became controllable, the exact alloy of the pipelinings to resist the corrosive backbite of the product. The 1,000 small intuitions that only years of handling these molecules could provide. Men who had served a regime of industrial murder became security assets because the weapon they had built was too strategic to leave to one superpower alone. The paperwork was careful. The scientists were given new identities, new laboratory coats, and new offices in the American desert. Gerhard Schrader, the man who had first smelled that faint almond scent on a warm afternoon in Leverkusen, never went to America. He stayed in Germany, returned to his pesticide research at Bayer, and lived until 1990, a pensioner in a tidy house, unprosecuted and unremarked upon, while the molecule he had drawn out of the air killed children in the streets of Damascus. The dismantling of what remained in the western zones proceeded under the supervision of Chemical Corps captains who inspected every flange and packing gland. Trucks loaded with fracture columns, mixing vessels, and sealed steel drums marked with the Anorgana logo bounced over the ruined roads of Bavaria toward French ports. The crates were stamped with consignment labels that read agricultural machinery or industrial solvents. At the quayside, dock workers sweated in the salty heat of Le Havre, heaving the boxes into Liberty ships that pointed their bows toward the Panama Canal. The journey through the tropics was a heat-drenched anomaly in the life of equipment designed for a northern forest. What the American captains did not know, could not know, was that Soviet intelligence was tracking the same crates through its own networks, photographing the same consignment labels from the dock at Le Havre. The dismantling of Die Herrenfurth was not a clean transfer. It was the opening move in a chemical arms race whose blueprint was shared, by accident of capture, between both sides. By the time the ships debouched into the Pacific and turned toward a quiet rail depot in the American West, the crates had arrived at their destination, a strip of prairie 18 miles northeast of Denver, where a new facility was rising against the flat horizon. At the Rocky Mountain Arsenal, under a sky so wide it seemed to absorb all sound, the U.S. Army Chemical Corps set about building a production line that would replicate and then surpass the German original. The first building was a near copy of the Die Herrenfurth main hall, but with thicker concrete walls, remotely operated valves, and a ventilation system that could exchange the entire air volume of the plant in minutes. The workers, many of them local hires drawn by the promise of steady pay, wore protective suits that grew bulkier with each passing year. First rubberized aprons, then fully encapsulated garments with sealed faceplates and piped-in air. At the end of a shift, they showered and changed into street clothes, stepping from a sealed world into the ordinary twilight of a Denver suburb. They were told the plant manufactured pesticides and specialty fuels, a story that fit the landscape, since the arsenal sat in the heart of the nation's agricultural belt, and the name of the product, they were told, was classified because of its commercial value. The cover held. For a quarter of a century, the true function of the nation's largest chemical weapon production site was a secret kept, in part, by its very ordinariness. In the decades that followed, the Denver suburbs crept steadily outward, new housing developments, shopping centers, schools, until the boundary between the secret city and the ordinary one shrank to a chain-link fence that passing motorists barely noticed. Families held barbecues in backyards less than 5 miles from the nation's largest nerve agent production line. They did not know. The cover held, as it had to, because the most effective camouflage for a weapon of mass destruction was not a classified document, but the unremarkable shape of an American suburb. The engineering problem that dominated those early Cold War years was not the manufacture of the agent. The German recipe worked with a quiet, terrible efficiency, but how to store it safely in peacetime. The liquid attacked seals built up pressure inside munition casings and was susceptible to slow, inexorable leaks. The original German plants had accepted a certain rate of worker exposure as a production cost. The American arsenal, operating under the oversight of a press and a Congress, could not. The solution the Chemical Corps devised was an elegant piece of chemical choreography, the binary weapon. The principle exploited a simple fact of the reaction. Sarin is formed when two separate chemical precursors, each far less dangerous than the final agent, are brought together under the right conditions. One precursor, a compound, the engineers designated DF, and another, a common alcohol called isopropanol, would be loaded into separate, physically isolated chambers inside the munition. A rupture disc between them would remain intact during storage and transport. Only when the artillery shell was fired, when the brutal acceleration of the launch broke the barrier and forced the two liquids into a mixing chamber, would the reaction begin. Inside the spinning supersonic projectile, the liquids churned and heated, and by the time the shell reached its target, the full agent would be present. If the shell never fired, it was merely a canister of industrial fluids, no more deadly than a drum of lacquer thinner. The lethal halo was deferred until the moment of use. By the early 1950s, the Rocky Mountain Arsenal was nearing completion. And here, the chapter's central contradiction edges into view. Adolf Hitler, who had ordered the industrialized killing of millions of civilians, who had shown no restraint in the waging of war, never authorized the battlefield use of his nerve agents. The reasons are still debated. Some historians point to his own experience of gas in the trenches of World War I. Others cite a flawed intelligence assessment that mistakenly assumed the Allies possessed similar weapons and would retaliate in kind. Still others argue that by 1944, the tactical window had closed. Allied air superiority would have made any German gas attack a suicide mission. Whatever the precise cause, the Führer's caution preserved millions of lives. And yet, exactly because the weapons were not squandered, they survived the Reich. The trophy that Hitler hoarded became the cornerstone of the very Western alliance that had destroyed him. The molecule passed from one empire to another, and its service life extended far beyond the man who had been too afraid to use it. But the weapon had been tested on human beings years before the first American shell left the assembly line in a secret laboratory in the English countryside in the spring of 1953. The chamber was a room designed to be forgotten. It had no windows. The walls were sheathed in metal and sealed with rubber gaskets, and the air that moved through it came from pipes that led to tanks that were never labeled in plain sight. A single bulb burned overhead, but the light felt thin, the way it does in a basement that has learned to keep secrets. The men who entered did so one at a time, wearing white coveralls that had been starched and pressed, as though the proper crease could hold back the thing they were about to meet. On a table inside the chamber, a glass dish held a clear, colorless liquid that smelled faintly of something sweet, or so a few of them said later. Most remembered nothing but the disinfectant. The year was 1953. The place was Porton Down, the United Kingdom's chemical weapons research establishment, set in the Wiltshire countryside behind hedgerows and official signposts that offered no hint of what the low brick buildings contained. For the young servicemen who arrived by lorry, it was presented as a posting like any other. Contribute to the nation's defense. Help the scientists understand how to protect a soldier against new threats. They were told they would be compensated for their time, that the experiments were safe, that they could withdraw at any moment. The word volunteer was used often, and it sounded to their ears like a fair transaction. A man gives his afternoon. The state gives its thanks. Ronald Madison was 20 years old, an aircraftman in the Royal Air Force. He had grown up in the industrial north of England, and he was, by all accounts, a quiet and ordinary young man who did not seek attention. When the call came for volunteers at Porton Down, he put his name forward. Perhaps he wanted to do something useful. Perhaps he wanted the extra pay, a few shillings that would look good in his pocket on leave. Perhaps he simply did what young men in uniform are trained to do. He said yes when the officer asked. On the morning of May 6, 1953, Madison changed into the white coveralls and entered the gas chamber. The protocol was simple. A technician applied a single droplet of a clear liquid to the bare skin of his forearm. The liquid contained a nerve agent the laboratory had been studying, Sarin, a molecule that had first been synthesized 15 years earlier in a German laboratory, but had since migrated, like so much of the Reich's unfinished business, into the care of the victors. The droplet was meant to test the effectiveness of protective clothing. Madison was wearing none on that patch of skin. He was, in the language of the experiment, the control. For a few minutes, nothing seemed to happen. Madison stood calmly while observers watched through a sealed window, their clipboards ready, their stopwatches ticking. The room was quiet except for the low hum of the ventilation system. Then the familiar cascade began. The physiological markers began appearing. Markers the observers had been trained to recognize, had seen in the lung tissue of laboratory animals, had read about in captured German reports. The stopwatches ran. The clipboards filled. Madison's body was not a patient, but a data stream. The observers noted the time. The onset had taken less than 3 minutes. Within 5, he was on the floor. The white coveralls no longer crisp, his body no longer responsive. The men behind the window had seen this sequence in animals, never in a human. The scientists at Porton Down were not amateurs. They knew exactly what the droplet could do, had seen it in animals, had studied its mechanism in reports captured from Germany and refined in their own laboratories. They had atropine ready, and they administered it. They tried artificial respiration. They worked on Madison for an hour, but the dose had been larger than any human body could absorb and survive. And at 11:15 that morning, Ronald Madison was pronounced dead. What followed is a study in the mechanics of institutional silence. The RAF informed Madison's family that he had died in a tragic accident. The coroner's inquest, held in secret, returned a verdict of death by misadventure, and the cause was recorded as asphyxia due to exposure to a poisonous substance. The precise substance was not named. The family was told that Ronald had suffered from a nervous condition, that he had been unwell before the experiment, that his death was an unforeseeable tragedy. They were forbidden from discussing the details. The official record was sealed, and for nearly 50 years, that was where the matter rested. A thin file in a locked cabinet, a name on a list that was not meant to be read. The strange part was not that the experiment had failed, but that it had succeeded in the one way the military most needed. Madison's death provided a data point. It established, in the starkest terms, a boundary of human tolerance. The droplet that killed him contained approximately 200 milligrams of sarin, roughly 10 times the lethal dose for an average man. Now the scientists knew, with terrible precision, what that number looked like in a human body. They knew how fast the symptoms appeared, in what sequence the organs surrendered, how long the window for treatment lasted before it became a window onto nothing. The death was tragic, but from the perspective of chemical weapons development, it was also extremely informative. In the corridor outside the chamber, the clock ticked toward midday. A clerk filed the day's incident report, the one with the official cause of death, under a classification stamp that would take 40 years to expire. The afternoon light of an English spring fell through the high windows onto a linoleum floor that had been mopped clean. No one who walked that corridor afterward had any reason to stop. Across the Atlantic, the same calculation was being made on a much larger scale. At Edgewood Arsenal in Maryland, From the early 1950s into the mid-1970s, more than 7,000 American servicemen were exposed to nerve agents in experiments known only to a small circle of researchers. They signed consent forms so vague the words could have covered anything from a headache to a heart attack, and often did. Soldiers entered test chambers in groups, wearing protective suits whose seams had been deliberately torn so researchers could measure the agent's penetration. Some collapsed. Some were evacuated to the base hospital, where physicians who had signed secrecy agreements treated them and carefully noted the dosages. The files described them as exposed subjects and their injuries as adverse events. The language did not flinch because it had been designed not to. The paradox at the heart of these programs is not difficult to state, though it was rarely stated inside the chambers themselves. The soldiers believed they were sacrificing for defense, but the weapons they were perfecting were not defensive. The suits, the atropine injections, the emergency protocols, they were afterthoughts, attempts to build a fence around a fire that had already reached the house. The real purpose of the testing was to measure how quickly the fire spread, how hot it burned, and at what distance it stopped being useful and started being a liability. The data was written in the language of medicine, but it was read by the language of war. Answers found in the bodies of young men who had been told they were helping to keep the peace. The smell inside the chambers was never the agent itself. Sarin is almost undetectable to human senses, a fact that became, in itself, a source of fear. The smell was the disinfectant, the rubber of the face masks, the metallic tang of the ventilation grates, and underneath it all, the faint salt of human anxiety. The volunteers learned to recognize their own fear by the way it sat in their stomachs, a cold, still weight that no amount of breathing exercises could shift. They would stand in their white suits, hands at their sides, waiting for the signal that the vapor had been released, and they would try not to think about the men who had come before them, the ones who had been carried out, the ones whose names they did not know. Ronald Madison's name, it turned out, would not stay buried. In 2002, a journalist named Rob Evans began to investigate the long-suppressed history of Porton Down's human experiments. He found documents that had been hidden from the Madison family for half a century. He found surviving volunteers who spoke of what they had endured. The case went to a new inquest, and in 2004, a jury returned a verdict that overturned the original finding: Ronald Madison had been unlawfully killed. The state had used him as a test subject without his informed consent, had exposed him to a lethal substance, and had then lied to his family about what it had done. The truth was delivered in a courtroom 41 years after his death. But the verdict, however just, did not close the file. The documents from Edgewood Arsenal remain, for the most part, in the custody of the Department of Defense. The thousands of American soldiers who were exposed have never received a formal acknowledgement of the full scope of what was done to them. The data they generated, the charts, the graphs, the clinical notes, is still cited in military manuals and toxicological reference works. It is knowledge acquired in a way that is hard to look at directly, and so the instinct is to look away, to let the fluorescent lights of the file room bleach the memory into something clean enough to store. But the body preserves its own evidence. The veterans of these experiments carry neurological deficits, chronic pain, respiratory illnesses, and a persistent, unanswerable question about the precise chemistry of their own past. They walk into routine medical appointments and discover that their blood work shows traces of a compound the civilian doctor has never heard of. They dream of rooms with no windows. They wake, decades later, still trying to identify the faint, sweet smell that lingered in the air before the world went tight. The gas chambers at Porton Down and Edgewood Arsenal are decommissioned now. Some have been demolished, others have been converted into storage spaces or training rooms, their history reduced to a line in a facility report. But the molecule that filled those rooms does not age. It does not decay. It remains what it has always been, a piece of chemical engineering so precise, so economical, that a droplet the size of a pinprick can collapse the nervous system of a grown man in less time than it takes to read a single page of the report that documented his death. The threshold was measured. The data was recorded. The chamber was sealed. And the question that was never answered, the question that haunted the white coveralls and the silent observation windows, is not whether the tests should have been done, but what kind of world we were building that made them seem necessary. The room is dark now, but the records are still there. The margin notes are written in a steady hand. The last entry in the logbook for May 6, 1953 does not mention a name. It simply notes the time, the dosage, and the outcome. The ink has faded, but it has not disappeared. The paper waits, in the half-light, for someone to ask what it knows. While the logbooks were being sealed, a different kind of test was already underway on the other side of the Atlantic. The first binary warheads were tested on the Utah salt flats under a sky as vast and blank as the Colorado sky. Detonation sent up plumes of white dust that mixed with the invisible vapor, and the engineers, observing through periscopes, measured the kill radius, the dispersal rate, the persistence on dry ground. The data was folded into the slow, dispassionate arithmetic of mutual assured destruction. The weapon that had been conceived in a German garden laboratory, then nursed to industrial scale by a regime of terror, had now been fully naturalized under the American flag. It sat in concrete bunkers from Alabama to Nevada, in the magazines of warships, in the bomb bays of long-range aircraft on round-the-clock alert. Its possession had become a national strategy, unmentionable and essential. One evening in the spring of 1962, a special train left the arsenal after midnight and crept through the mountain passes toward the Pacific. A single steel container, no larger than a domestic oil tank, was offloaded and carried into the belly of a B-47 bomber that took off before first light. The bomber flew long circles over the desert, its crew waiting for an order that never arrived. The mission was a rehearsal, one of hundreds that tested the choreography of chemical warfare. The plane returned to base, the container was removed, and the entry in the flight log was stamped with a classification that would not expire for 50 years. The weapon had not been used. It had simply been moved, in readiness, from one sealed vessel to another. The silent machinery of the captured plant, dismantled and crated for shipment to the American desert, had been reborn as a production line that would run for 3 decades. The knowledge it carried was embedded in a continent-spanning architecture of deterrence, not one arsenal, but 7, not one nerve agent, but 5. And yet the ordinariness of the weapon's storage, the routine pressure checks, the weekly inventories, the budget lines that appeared in congressional hearings under headings no one questioned, gradually dulled the horror that had first surrounded it. A weapon that can be managed becomes a tool. A tool can be imagined in limited use. The Cold War never saw sarin deployed against a city, but that is only the fortunate half of the history. The other half, the small wars, the proxy battlefields, the basement laboratories of cults, would prove that the trophy of the Reich had a way of escaping the plans that empire laid. The first thing Reich noticed that morning was the stillness. The wind that usually scraped across the salt flats west of Salt Lake City had gone somewhere else, leaving the valley under a blue dome that seemed to hold its breath. He loaded his pickup and drove out to the winter range just after sunrise on the 13th of March, 1968. The land was a checkerboard of government grazing allotments and private holdings, stretched beneath the Cedar Mountains. Peck ran about 4,000 sheep. The week before, a cold snap had pushed some of them down from the higher ground. But today the weather was mild, the sky cloudless, the kind of morning a rancher learns to trust. He found the first ones near the fence line. A ewe lay on her side, legs stiff, eyes wide. A few yards farther, a dozen more collapsed as if they had been dropped from a height. No blood, no sign of a predator, no tracks in the dust. While Peck stood beside his truck, a lamb stumbled toward him, fell, and went into convulsions. The high desert air, so often sharp with salt and sage, carried an odor he could not place. Not quite chemical, not quite organic, something that irritated the back of the throat. By the time he could get to a telephone, he had counted more than 100 dead. That same morning, across the valley and into the afternoon, other ranchers were discovering the same thing. The phones at the Skull Valley Livestock Association started ringing. Within 12 hours, the number of dead sheep climbed past 2,000. Within 24 hours, 6,400. Entire bands had been wiped out. Ewes, lambs, rams, animals that had grazed these winter ranges for generations lay in windrows across the alkali soil. The scene was so strange that the local veterinarian sent samples to the state pathology lab, suspecting poison. The lab found something stranger. The sheep had died of a nervous system collapse, typical of anticholinesterase exposure, but no agricultural pesticide in use at the time could have produced it. The blood and tissues held a signature that looked, to the trained toxicologist, like a chemical designed for war. 60 miles to the west, hidden behind the Granite Mountains, the U.S. Army operated the Dugway Proving Ground, nearly 800,000 acres of salt desert, where ordnance, biological agents, and, in an especially guarded section, nerve agents were tested. In the spring of 1968, Dugway was one of the most secret places on the continent. It was here that the stockpiles of American chemical weapons were brought for open-air trials. It was here that the chemistry born in a German laboratory 3 decades earlier was measured against the desert wind to study how an invisible cloud of sarin would dissipate, drift, and kill. On the morning of March 13, a routine test flight had taken off from Dugway's runway, a modified cargo aircraft carrying a spray system designed to release liquid sarin into the atmosphere for meteorological tracking. A valve on one of the dispensing tanks failed. Instead of cutting off at the designated altitude, it leaked roughly 9.5 gallons of liquid sarin into the slipstream. The agent aerosolized, caught the air currents that Reich had noticed were missing, and rode them silently over the mountains and onto the valley floor. The weapon had no politics. It moved with the same physics as the weather: temperature gradient, wind shear, atmospheric boundary layer. But the people who received it had everything to lose. By evening, the spotter planes that flew for the army were reporting dead birds, dead rabbits, dead coyotes across the public lands outside the test zone. But the central mass of the plume had settled onto the winter range, a shallow basin cool enough to hold a heavy vapor. The sheep, breathing close to the ground, took the full dose. The ranchers, living a few miles away, were sickened but spared. The invisible cloud that was meant to stop a Soviet division had proved, instead, that it could not distinguish between a tank column and a flock of sheep. Its precision was an illusion of the laboratory, a paper calculation that the wind erased in the space of a morning. The Army's first public statement was a denial. There was no admission of malfunction. Officials suggested the sheep might have been killed by an organophosphate pesticide from an agricultural source, or by a natural outbreak of disease. The ranchers, with Peck among the most insistent, did not believe it. They could see the flight logs, the strange spotter aircraft circling the day before. They knew the wind had shifted. In the weeks that followed, investigators from the veterinary laboratory and the Department of Agriculture identified residues of sarin breakdown products in the soil and the dead animals' tissues. The chemical fingerprint was unambiguous, a set of phosphorus-bound metabolic fragments that matched exactly the degradation pathway of GB, the military designation for sarin. Still, the Army resisted. It was only after a year of litigation and mounting scientific evidence that the government acknowledged liability. In 1969, the Army paid the ranchers $376,000 in damages, a small figure against 6,400 dead animals, but an enormous figure in the calculus of secrecy. The payment meant admission. The admission meant that the American public was learning for the first time that the United States was not only stockpiling chemical weapons, but testing them on the wide open landscapes of the interior West. The rows of dead sheep lying on the salt flats became a different kind of evidence. No longer a military secret, they were photographed, counted, and cataloged by the press. The images traveled to newspapers in Salt Lake City, then to wire services, then to foreign capitals. For a public that had come to think of nerve gas as an abstraction, a Cold War rumor, something that existed only on the other side of the world, the sight of American-bred livestock crumpled on American soil was a shock that rewrote the cost of deterrence. The poison had drifted less than 30 miles from the proving ground to the pasture. It had crossed no border except the one between government secrecy and private life. In the aftermath, the Dugway incident forced the hand of the chemical weapons program. Congress held hearings. The open-air testing of nerve agents was suspended, though not before other accidents had already been buried in classified reports. The safety of the weapon, as the engineers had designed it, was predicated on a level of control that the physical world did not offer. A valve could fail, a wind could shift. A morning that began with a rancher checking his flock could end with a map of dead zones invisible until it was too late. The political pressure from Skull Valley reached the White House. In November 1969, President Richard Nixon announced the unilateral renunciation of first use of lethal chemical weapons and ordered the halt of their production, a decision shaped, in part, by the image of a Utah valley carpeted with dead livestock, and by the recognition that the cost of deterrence, when measured in the currency of public trust, had become too high. To Ray Peck, the settlement check was the end of a season of grief and the start of a longer anger. He had seen his livelihood, animals he had raised from birth, become a laboratory trace, a column of numbers in an Army incident report. The money compensated him for the market value of the sheep, but it could not restore the trust he had held in the land itself. The valley looked the same after the spring rains, but Peck told reporters that he never again walked those hills without thinking about what was left in the soil, what the next wind might carry, and whether the government was still testing something he wasn't supposed to know about. the archive of the chemical weapons program grew a new file. It was labeled with coordinates, toxicological assays, and legal settlements, but it contained a deeper record, the first significant public awareness that the dangerous margin between a military test range and a civilian home was not a fixed line. It was a function of wind speed, air temperature, and the decision of a pilot who believed his valve was closed. The rows of dead sheep on the salt flats had no military classification. They were simply a visible, undeniable answer to a question nobody had thought to ask. What happens when the most precisely engineered poison falls from the control of the men who built it? The pure, empty blue sky gave no answer. It just held the light as the carcasses were loaded onto flatbeds and driven away. The wind returned the next day, as it always does, and swept the valley floor clean of any color but white salt and gray-green brush. The spring lambing season arrived and produced only sorrow. The summer drought sealed the bones. By autumn, the Skull Valley was, to any passing observer, an ordinary corner of the rural West again. But a new awareness had lodged itself in the public mind, an understanding that the Cold War's deadliest machines were not housed only in missile silos or under the ocean. They were breathing the same air. The lesson of Skull Valley was a local one. A single accident, a single compensation payment, a single valley that would carry its buried history in silence. But the same strategic logic that had driven the arsenal at Rocky Mountain was now reaching far beyond the borders of the Cold War superpowers. Chemical weapons had become a currency of power that even the smallest state could acquire, through technology transferred under the banner of aid, through the sale of precursor chemicals labeled as agricultural inputs, through the quiet complicity of nations that looked away. The lesson of the sheep would be rewritten on a different scale, in a Kurdish town that the world had chosen not to see. The cassette arrived at the London Bureau late on the evening of March 17, 1988, sent over a satellite link that turned the image into a shifting mosaic of gray and brown. When the tape was played on the newsroom monitor, the first thing the editor noticed was the silence. There were no crowds, no ambulances, no shouted orders. The street in the Kurdish town of Halabja was filled with people who were no longer moving. A man lay crumpled against a wall, his arms still reaching toward a door he had never reached. A woman had fallen with a child curled against her chest, their clothes unmarked. Further along, a group of bodies clustered near the entrance of a house, as if the family had tried to run out into the open air and had collapsed before they could take 3 steps. The camera tilted up, and the sky over the mountains was a pale, indifferent blue. Halabja sits in a valley in northeastern Iraq, close to the Iranian border. In March 1988, it was a town of roughly 80,000 people. That morning, the bread ovens had been lit before dawn, and children had walked to school along streets that smelled of wood smoke and diesel. The war between Iran and Iraq had been grinding on for 7 years, but the town itself had been spared the worst of the frontline fighting. People were accustomed to the distant thud of artillery and the occasional rumble of aircraft. At 11:30 in the morning, a low-flying squadron of Iraqi Mirages and MiGs appeared over the ridges to the west. They did not bomb the military targets that were not there. They released canisters that fell slowly, trailing a yellowish-brown mist that drifted downward with the air currents. The fog pooled in the alleys and crept under doorframes. It carried a scent that some survivors described as garlic or rotten fruit, but others remembered nothing at all, because the nerve agents it contained began to attack the body before the brain could register an odor. The mixture was a laboratory cocktail, Sarin for its speed of action, Tabun for its persistence in the environment, VX to penetrate intact skin without the need for inhalation, and mustard gas to blister the lungs and blind the eyes. The yellowish-brown color was not the color of sarin. The pure nerve agent is colorless, but the signature of a deliberate blend designed to maximize terror across every route of exposure. The Iraqi Chemical Corps had spent 5 years perfecting its recipes on the battlefields of the Iran-Iraq front, and Halabja was the first time the full menu was served to a purely civilian population. In the space of a few hours, more than 5,000 people died. Those who survived would carry the physical signature of that exposure for the rest of their lives. In the streets of Halabja, the dust settled on a silence that no breeze disturbed. A dog lay curled on a doorstep, its legs frozen in the motion of a run it had not finished. The cassette tape in the camera continued to turn, recording a stillness too thorough to be mistaken for peace. Then the camera operator lowered the viewfinder, and the ordinary machinery of international reaction began its slow, deliberate course. The chain of statements, the diplomatic consultations, the filing of reports that took care to use the conditional tense. The attack was not an isolated incident. It was the deadliest stroke in the Anfal campaign, a series of military operations designed to depopulate Kurdish areas. Between February and September 1988, Iraqi forces launched multiple chemical strikes against villages and towns. Halabja was the largest, but the pattern was systematic, and the campaign had been enabled by a quiet network of international trade. The Iraqi chemical weapons program did not emerge from a secret underground bunker. It was assembled, piece by piece, through the global chemical market. Western companies in West Germany, the Netherlands, France, the United Kingdom, and the United States had sold Iraq the precursor molecules and the specialized equipment needed to build pesticide factories. A Dutch firm shipped thiodiglycol, a mustard gas precursor labeled as textile softener. A German firm provided the glass-lined reactors. A British company exported hydrogen fluoride, a compound used to produce sarin. The shipments were cleared by export control agencies that chose not to see the destination's ultimate purpose. The knowledge was not stolen, it was invoiced. When the United Nations Security Council first debated Iraq's use of chemical weapons in 1983, the evidence was already overwhelming. Satellite imagery showed the craters of chemical munitions. Defectors had brought samples. Doctors had documented the characteristic symptoms. But the Reagan administration instructed its diplomats to block or weaken any resolution that might embarrass Baghdad. The calculation was geopolitical. An Iranian victory on the battlefield was seen as a greater threat than an Iraqi chemical arsenal. Iran was the designated enemy, and the Kurds were the collateral. This was the quiet engine behind the massacre. The Geneva Protocol of 1925, which prohibited the use of chemical and bacteriological weapons in war, had been ratified by more than 100 states, including Iraq. But the protocol had no enforcement mechanism, no inspection regime, and no penalty for violations. It was a law made of words, and the words had been carefully chosen to avoid interfering with the sovereign right of states to produce whatever chemicals they wished. In the decades after the First World War, the great powers had preferred a weak ban to a strong one, because a strong ban would have revealed the size of their own stockpiles. The result was a legal instrument that looked like a wall on the map of civilization but dissolved into air the moment a state decided to walk through it. In the immediate aftermath of Halabja, the State Department issued a statement expressing deep concern and urging both parties to refrain from chemical weapons use. The The formulation was a deliberate untruth. Iran did not possess a chemical arsenal. The sentence treated a genocidal attack and a defensive war as symmetrical because symmetry was the language of diplomacy and diplomacy was the servant of policy. The United Kingdom and France adopted similar postures. No ambassador was recalled. No sanctions were proposed. The details of the massacre were consigned to the back pages of newspapers, and the war continued. The Kurdish dead became a footnote in the history of the century's last great conventional conflict. But the poison did not conclude its business with the dead. In the months and years that followed, the survivors began to report a new map of illness: miscarriages, stillbirths, children born with brains that had not closed, with limbs that had not grown, cancers that appeared in clusters, leukemias that struck toddlers, lung tumors in non-smokers. The pattern was so consistent that local doctors began to speak of a chemical generation's effect, but they lacked the resources to prove it. The world's medical establishment, absorbed in other catastrophes, paid little attention. Dr. Christine Gosden was a professor of medical genetics at the University of Liverpool, a specialist in the hereditary patterns of disease. In 1991, she saw a series of photographs from Halabja in a medical journal. What disturbed her was not only the images of the dead, but the complete absence of any systematic follow-up on the living. She had spent her career tracing the footprints of genetic damage through families, and she understood that an event like Halabja was not over. It was a slow, burning epidemic disguised as a political memory. In 1995 and 1996, she traveled to the refugee camps and the ruined quarters of the town. She interviewed hundreds of survivors, gathered blood and urine samples, photographed skin lesions and eye damage, and documented the medical histories of entire kinship networks. What she found, and later published in a series of peer-reviewed papers and parliamentary testimonies, rewrote the textbook on chemical weapons casualty estimation. The standard military model had always focused on acute lethality. How many people die in the first 24 hours? Gosden's data showed that the chronic burden was orders of magnitude larger. The cancer rates in the exposed population were up to 20 times the expected levels for certain types. Birth defects occurred in terrifying clusters: anencephaly, spina bifida, cleft palate, limb reductions. Psychiatric illness, severe depression, post-traumatic stress disorder, psychosis was endemic. The epidemiological curve did not fall after 5 years or 10. It continued to rise, generation after generation, because some of the chemical agents had damaged the DNA in the germ cells of the survivors. The poison had not just scarred the bodies of the parents, it had written a new instruction into the genetic heritage of the community. The biological mechanism was not wholly understood. Mustard agents are alkylating chemicals. They can bind to DNA and cause mutations that are inherited. The nerve agents, by disrupting the neurotransmitter acetylcholine, may interfere with the delicate signaling that guides fetal development, producing structural abnormalities that mimic genetic defects. In one family she studied, a grandmother exposed in 1988 had given birth to a daughter with severe learning disabilities. That daughter, in turn, had a child with a neural tube defect that required lifelong care. The grandmother's sister, who had been visiting relatives outside Halabja during the attack, had given birth to 3 healthy children. The contrast was written not in policy documents, but in the flesh. Gosden called the people of Halabja the canaries in the mine, the early warning system for a class of weapons that did not end with a ceasefire. Her work was meticulous, dispassionate, and it presented a picture of chemical warfare that no military doctrine had prepared for, a weapon that outlives the battlefield and settles into the very blueprint of human life. The Geneva Protocol had nothing to say about the slow death. Neither did the Chemical Weapons Convention, which was still under negotiation in the early 1990s and would not come into force until 1997. The convention would eventually outlaw the production and stockpiling of nerve agents, but even its strongest provisions could not undo the past. Halabja demonstrated that a chemical atrocity left a legacy that no treaty could erase. It also demonstrated that a treaty without commitment from the powerful was a sheet of paper. The same nations that had sold the ingredients and looked away in 1988 would later cite Halabja as a reason to strengthen the CWC, as if denunciation could retroactively supply the political will they had withheld. In a small clinic on the outskirts of Halabja, Dr. Gosden examined a 7-year-old boy whose mother had been pregnant during the attack. The boy had never walked. His legs were twisted, his eyes tracked poorly, and he suffered from uncontrolled seizures. The mother kept a photograph of her family taken the year before the planes came. In the photograph, she stands with her husband and their 2 older children, all of them smiling, the valley green behind them. The boy was not in the picture. He was the child of a different world, the one that began at 11:30 on March 16, 1988. Dr. Gosden placed the photograph back on the shelf and continued her questionnaire. The record she was building was not for the boy. It was for the archive of a crime that the law had chosen not to see. The last reel of the news footage from that week ends with a slow pan across a street of rubble. The camera pauses on a child's sandal, small and dusty, one strap broken. It is the kind of object that washes up in all massacres, the ordinary things that become unbearable. The sandal does not tell you the name of the child or whether the child survived. It tells you that the morning had begun normally, that someone had buckled the strap and set out for the day, and that the fog arrived before the day could finish. The sandal is still there, in the archive of the television station, filed under Kurdistan, 1988. The cassette sits in a cool, dark room alongside thousands of other cassettes, waiting for the next time the world decides to remember. The poison itself is not on the tape. It is in the soil, in the water table, in the cells of the living, and in the clauses of a treaty that has yet to be enforced against anyone who matters. The poison in the soil, in the water table, in the cells of the living, this was the legacy of a precedent the world had chosen not to enforce. A generation later, that legacy would surface again, in another valley under another sky, when the weapon that had fallen on Halabja was turned on a suburb of Damascus. The cell phone video begins in the blue hour before dawn. The screen trembles. The person holding it is running. In the frame, a field hospital corridor, bodies on the floor, a man in a white coat moving too fast. The sound is a composite of gasps, Arabic curses, and the wet rhythm of a suction pump. The date is August 21, 2013. The place is Ghouta, a suburb of Damascus under opposition control. The sun has not yet risen, but the weapon has already done its work. The images showed the aftermath of a nerve agent attack, but the context, a civilian suburb, a morning like any other, was entirely new to the international news cycle. The weapon was not a secret anymore. It was live-streamed. The weapon was Sarin. It had arrived in metal canisters, delivered by surface-to-surface rockets, timed to hit when families were still in their homes. The windows closed against the pre-dawn cold. By the time the video reached the world, the dead numbered in the hundreds, the injured in the thousands. The event was not a laboratory accident, not a cult's paranoid vision, not a battlefield in a war between states. It was a government gassing its own population in a conflict that had already blurred every distinction between combatant and civilian. The contradiction was immediate. The attack occurred in a world where an international treaty had already banned the weapon, and where a verification system was supposed to ensure compliance. A.K. Sellström arrived in Damascus shortly after. He was a Swedish scientist, a chemical disarmament veteran with a quiet voice and a methodical mind. His task was to lead the United Nations mission to investigate the allegations. The mission's white land cruisers moved through the capital under Syrian military escort, their path negotiated checkpoint by checkpoint. Inside the vehicles were sampling kits, personal protective gear, and the dry, practiced patience of inspectors who understood that access was a currency the host state would dispense in small denominations. The investigators were not welcome, they were merely tolerated. Sellström had done this before. He had walked through other ruins, collected other samples, written other reports that had been filed and forgotten. What stayed with him was never the chemistry. The chromatography was routine. The chain of custody paperwork, tedious but reliable. What stayed was the gap between what he could prove and what he could see. The mandate was to establish whether chemical weapons had been used, not who had used them. That line, assigned in advance by a Security Council fractured by the Syrian war, was a line drawn through the map of responsibility. In the evenings, Sellström reviewed the sample logs, soil from impact craters, clothing from victims, blood and urine from survivors, and understood that each vial held a truth that the mandate would not allow him to complete. The site visits themselves were a study in contradiction. The inspectors moved through the ruined streets of Ghouta in their white coveralls, taking photographs, marking coordinates, sealing evidence in tamper-proof containers. The local population watched them with the exhausted eyes of people who had already seen too many delegations arrive and leave without consequence. In one laboratory later, a chemist who examined the soil samples noted a faint residue that matched the synthetic pathway of military-grade sarin. The molecule had left its mark, as it always did, an industrial signature that could only have come from a state-run chemical weapons program. The precise formulation was not the crude mixture of an amateur. It was the optimized product of a dedicated chemical weapons program. The international response was shaped by a year-old statement. President Obama had warned in 2012 that the use of chemical weapons would cross a red line and trigger serious consequences. Now the line was crossed, and the footage was public. The diplomatic machinery turned. Within weeks, Russia and the United States brokered a deal. Syria would join the Chemical Weapons Convention, declare its stockpile, and allow the removal and destruction of its chemical weapons. The world, in the language of the time, had chosen diplomacy over missiles. The declared chemicals were loaded onto ships at Latakia in 2014, a marine convoy that sailed into the Mediterranean under the eyes of international observers. The treaty, it seemed, had been vindicated. A stockpile was gone without a bomb dropped. But the treaty operated on a register of declarations. It was a voluntary compact, a promise made by states to reveal what they possessed. The verification system was designed to confirm that a declared stockpile was accurately described and thoroughly destroyed. It was not designed to search for arsenals that a state denied having. The inspectors were not detectives, they were auditors. They could only check the books that were placed before them. If a country chose to keep a second set of books, or to move a production line into an unregistered building, to bury precursor chemicals under a warehouse floor, to hide munitions in a cave outside the declared perimeter. The treaty could not see it. Syria sent its declaration to The Hague. The OPCW verified it. The declared chemicals were destroyed, but no one could prove the list was complete. The treaty had no mechanism for proving a negative. The silence of an undeclared stockpile was indistinguishable from the silence of an empty room. In April 2017, that silence broke. The town of Khan Sheikhoun in Idlib province was hit by an airstrike that released sarin. The attack killed dozens, perhaps over 100, and the video footage returned to the screens of the world. The OPCW fact-finding mission, the institutional descendant of Sellstrom's team, deployed again. Their report confirmed sarin exposure and noted that the sarin used was chemically similar to the samples from Gouda. The conclusion was inescapable. The destruction of the declared stockpile had not eliminated the weapon. Undeclared sarin had been retained, hidden from the treaty's gaze, and deployed when the tactical need arose. The treaty's successful removal of tons of chemicals had been a triumph of diplomacy, but it had also been a decoy, a transaction that convinced the world to look away while the real arsenal rested in the dark. The cell phone videos from Gouda are still retrievable. They exist on servers, in forensic reports, in the memory of the networks that first broadcast them. A viewer who watches them now, a decade later, will see the same convulsions, the same rush of medical personnel, the same frantic voices. The videos do not age, but the political context around them shifts. New attacks occur, new investigations launch, new reports are filed. The Chemical Weapons Convention remains the law of the land, with almost all its state parties in compliance with their declarations. But the undeclared are not parties to that statistic. The weapon, in its molecular form, does not age either. Its old 50 is unchanged. Its volatility is the same. The only thing that has changed is the expectation that the treaty will, by itself, keep it from the lungs of children. That expectation, on the morning of August 21, 2013, proved to be a frail instrument, easily shattered by a rocket's impact in the dust of a Damascus suburb. The white UN vehicles are long gone from Syria, replaced by other missions, other emblems, other white vehicles in other conflict zones. The sealed evidence containers rest in the OPCW laboratory archive in Reich near The Hague. They are kept cold and dark, samples labeled with the date and coordinates of their collection. A soil jar from Ghouta, a blood vial from Khan Sheikhoun. The samples are not dangerous. The sarin has degraded into its phosphonic acid residues, the chemical echo of a molecule that has done its work and vanished. The treaty, in its bound volume, is similarly dormant, a set of words that depend entirely on the will of its signatories to become action. The ghost in the protocol is not the sarin itself, but the presumption that a signature is the same as a confession, and that a confession is the same as a renunciation. The ghost lives in the gap between what is declared and what is denied, and that gap is not closed by law. The last thing the investigator remembers is not a chemical formula, but a smell. The air in Ghouta after the attack had an acrid quality, the stale perfume of explosive residue mixed with the unmistakable scent of mass death. But beneath it, there was the cool, plain air of a winter morning in the Levant, the same air that had always passed through those streets in peacetime. The sarin had no scent. It could not be smelled. It had moved through that ordinary air, invisible and indifferent, and then it was gone, leaving only the bodies and the videos and the laboratory reports. The treaty had promised to make the world safe from such a weapon, and for a time, in the conference rooms of Geneva, the promise had felt substantial. But the chemical threat had migrated from the laboratory bench to the factory floor, from the superpower arsenal to the cult compound, from the declared stockpile to the undisclosed depot. Each migration had found the law one step behind, writing the rules for the last disaster, while the next was already underway. The catalog of what remains to be faced is not closed, and the next entry is already being drafted in the silence of an undeclared laboratory in a place that the treaty does not know. 2 decades after the subway attack, Dr. Nobuo Yanagisawa kept a small, windowless room on the 3rd floor of St. Luke's International Hospital. The room was not for patients. It was for the long memory of the institution, a repository of gray metal cabinets, temperature logs, and specimen jars lined up on steel shelves like minor reliquaries. One jar held a single drop of inert serum drawn from a survivor in the weeks after the exposure. The serum had been centrifuged, separated, and stored in a buffered solution that kept its proteins suspended in a pale amber translucence. The drop was odorless and still. Under the fluorescent lights of the archive, it looked like nothing at all. A bead of condensed breath on cold glass, a spill that had already dried. But the file that accompanied the jar was thick with years of follow-up notations, and those notations told a different story. Dr. Yanagisawa had spent the decades after the attack tracking what the body does with a poison it never fully clears. The acute phase of organophosphate poisoning was brutal but legible. The regulatory systems of the body overwhelmed by a signal that would not stop. What came after was quieter and in many ways more unnerving because it blurred the line between toxicology and ordinary decline. Survivors reported persistent memory disturbances, a cognitive fog that settled into their working days and never lifted. Some described a numbness in the hands and feet that arrived without warning, as though the nerves had forgotten how to report pressure. Others experienced visual distortions, halos around streetlights, a fluttering at the edge of vision that came and went for years. The symptoms were not dramatic enough to make headlines, but they were durable. They turned a single morning commute into a chronic condition. The strange part was not that the damage lasted. The strange part was that it sometimes appeared to deepen long after the agent itself had been metabolized. The acute poisoning had ended within hours for most who received treatment. The molecule itself had been cleared or had aged into an irreversibly bound complex that the body slowly replaced. Yet the neurological complaints kept drifting into the clinic years later, like late, arriving mail from an address the patient had forgotten. Yanagisawa's team documented the pattern in careful clinical prose, but the prose could not resolve the underlying question, whether the weapon had merely wounded the nervous system or had reprogrammed it in ways that unfolded over time. The files stacked in the archive did not offer a verdict. They offered a population of people living inside an experiment they had never consented to. Half a world away, in the Kurdish regions of northern Iraq, a parallel archive was being assembled from the survivors of Halabja. There, the exposure had been heavier, the chemical cocktail more complex, and the medical infrastructure almost non-existent in the immediate aftermath. But the long-term toll followed a similar silhouette: chronic respiratory illness, elevated rates of neurological disorders, and a particularly cruel inheritance in the form of congenital abnormalities and developmental delays among children born years after the attack. The molecules had entered the germline indirectly, not by mutating DNA in the classical sense, but by reshaping the maternal physiology, the endocrine environment, the placental interface, the subtle biochemistry of gestation in ways that altered fetal outcomes. The bodies of the survivors had become archives of a different kind, passing the record of exposure into the next generation without a single gene having to change. The weapon, in this sense, did not need to be used again to continue its work. In March 2018, the lineage extended itself once more in the quiet English city of Salisbury. A former Russian intelligence officer, Sergei Skripal, and his daughter Yulia were found slumped on a public bench, incapacitated by a nerve agent that investigators soon identified as a Novichok compound, a fourth-generation organophosphate developed in the Soviet Union during the closing decades of the Cold War. Novichok was not Sarin, but it belonged to the same chemical genealogy, a branch of the family tree that had been cultivated in secret laboratories while the world was congratulating itself on the Chemical Weapons Convention. The agent had been smeared on a door handle. A few weeks later, a discarded perfume bottle containing the same substance killed a local woman, Dawn Sturgis, who had no connection to espionage. The incident demonstrated what the Tokyo survivors already knew. The weapon does not recognize its target. It diffuses along the ordinary pathways of daily life: a subway train, a park bench, a door handle, a bottle found by chance, and punishes anyone who happens to be in the radius of its patience. The doctrine of dual use, so often invoked in arms control seminars, is usually described as a policy problem. The same precursor chemicals that go into pesticides also go into nerve agents. The same fermentation vats that make pharmaceuticals can be reconfigured for toxins. But the deeper truth is that dual use is not a loophole to be closed. It is a property of the molecule itself. A phosphorus carbon bond does not know whether it belongs to an insecticide or a chemical weapon. The distinction exists only in the ledger of human intention, and intention is the most fragile container ever devised for a dangerous compound. You can write laws, erect verification regimes, and destroy declared stockpiles. You cannot write the synthetic pathway from the literature. You cannot make chemistry forget its own possibilities. That was the uneasy equilibrium that settled over the world after the Chemical Weapons Convention entered into force. Sarin was listed on Schedule 1, the most restricted category, alongside its cousins Tabun, Soman, and VX. The treaty mandated the destruction of all declared stockpiles within fixed deadlines, and most state parties complied, not without delays and cost overruns, but with a grinding bureaucratic momentum that eventually emptied the munitions depots and incineration plants. The samples that remain today exist only in a handful of state party laboratories, stored in milligram quantities for the purpose of verification analysis. They are sealed in glass, logged in double entry ledgers, handled under the kind of security usually reserved for pathogens and fissile materials. They are supposed to be the last physical traces of a weapon that the community of nations has collectively renounced. And yet the treaty itself contains a paradox that no draft language could resolve. The small, legitimate samples kept for testing are chemically identical to a seed stock. The forensic methods developed to detect illicit production also constitute a curriculum for anyone who wishes to understand how the molecule behaves. The OPCW inspection regime is one of the most intrusive and technically sophisticated verification systems ever invented by international diplomacy, but it relies on state declarations. A nation that simply refuses to declare, or that declares falsely, can keep a production capability hidden for years, as Syria demonstrated. The destruction of the world's declared sarin stockpiles was a triumph of law over logistics. It was not a triumph over the periodic table. Dr. Yanagisawa eventually closed the last of the long-term follow-up files. He had done what a clinician can do, documented the damage, prescribed what relief was available, and published the results so that the medical community would have a baseline for the next time. The serum sample in the archive was a relic of the past, quiet and harmless. But he understood that the archive was never truly closed. Somewhere, at any given moment, a chemist with the right precursors and the wrong purposes was recreating a synthesis that had been published for the first time in a German patent application in 1940. It did not age, it did not tire, and it did not distinguish between the Cold War and the morning commute. It waited in its own quiet way, a single drop of pale liquid, smaller than a coin, suspended in a glass vial in a locked cabinet for the next laboratory to open its doors. The question was not whether the weapon had been banished. The question was whether the world had learned enough to notice the door before it opened. The serum drop sits in its jar, amber and still, a bead of condensed memory from a morning that never ended. 2 decades after the Tokyo subway, it holds nothing dangerous, just the pale protein trace of a body that once met an invisible molecule and kept breathing. But the file beside the jar is thick with years of follow-up notations, the chronic headaches, the numbness in the fingers, the children born with anomalies that the poison did not directly cause but somehow preceded. The drop is evidence of what the body cannot forget. Across the salt flats of Skull Valley, the sheep still graze, but the bones of 6,400 animals lie sealed in the soil, a geological record of a valve that failed on a spring morning. In the forensic chromatograms from the cult's laboratory, the byproduct peaks rise like fingerprints signed in carbon and phosphorus, proof that a hasty synthesis leaves a trace even when the chemist intends none. And in the cell phone video from Gouda, the blue hour before dawn carries the same unlearned lesson. The molecule is indifferent to borders, but it always finds the lungs of someone who never chose to meet it. What became visible through these descents is not simply the history of a weapon, but the distance between invention and consequence, a gap that no treaty, no verification regime, no act of diplomatic destruction has ever fully closed. The knowledge of how to assemble this configuration of atoms does not degrade. It waits in the open literature, in declassified patents, in the memory of chemists who believed they were protecting crops or winning wars. The useful thing, the molecule that could kill a pest or a man, carries a hidden cost that continues to be paid long after the factory gates close or the arsenal is incinerated. The archive of what was done with that knowledge is not finished. It is only, for now, what we have recovered from the deep. The pressure eases. The darkness thins from black to blue. The hatch opens, and the cold sea air fills the lungs, not the air of a subway car or a desert testing ground, but the air of return, carrying no residue. If this kind of quiet descent into science and history is what you came for, you can subscribe to The Deep Archive. The serum drop remains in its jaw, inert and waiting. That is where the truth stays, between the locked cabinet and the next hand that opens it.